I also talk something about virus sanitation, which we can also use some new tools for that. So it's a whole lot, and I will hope I can keep your attention. The first we start with just showing a little bit very short. My university is in Belgium, surrounded by France, Germany, and Holland, and also England. And it's in Ghent that we are. Ghent is, I don't know if you can see my cursor moving, or should I?
Yes, we can.
Okay, good. Ghent is it's a medieval town with a lot of students, of course, from our university. And let's start now this topics. The first topic we will start is micropropagation. Short overview, of course. First, I don't know all your background, but micropropagation is about cloning the best plants. That's the meaning, the best tree, the best flower, the best fiber plants, and so on. And it all starts but one shoot, and thanks to the cytokinins, we can make that shoot growing and releasing its apical dominance, and then it's making a lot of shoots. I sort of split them here. It's 10 shoots. It's a lot here. It was for demonstration to show very quickly how we can multiply every 6 weeks by a factor 10. Or let's say here we can produce millions of plants in 1 year. Every 6 weeks is times 10, 10, 100,000, 10,000, 100,000. And what is propagated here? It's a tree from Kenya, Melia volkensi. We worked a long time on this tree. It's savanna tree, which was completely poached, let's say, by the Kenyans because of its very precious hardwood, mahogany wood, and it was only extinct, yeah. And it was luckily saved by some initiatives, and now it's planted. So I think 3 million trees are planted now at this moment of this nice tree. And we're looking also for a way to propagate the best trees of this the plant. So there's no, sometimes they say propagation 1 million plants, that's not good for the biodiversity. Okay, but there is biodiversity, 3 million seedlings. And now we are in the phase that we pick out the best of those 3 million to propagate the clonal woods, clonal forestry, the best clones. So you can propagate them, then they have to root it. The next step is rooting and then acclimatization into the nursery, and then the planting. And they grow very fast, those trees. It is expected that they can be harvested after 20 years. And it's extremely nice wood, termite resistant, and so on. So this is worthwhile to do this micropropagation, but of course there are much more plants to micropropagate, from the food plants, fiber, feed, also for fun, and so on, pharmacy, all the Fs. And the aim is, of course, to produce those plants in small recipients, and that they take on like a miniature habit, yes, and that you can grow them in small spaces, sterile, and so on. For the cytokinins are the key for this facet of micropropagation, of in vitro culture, the micropropagation. There's two ways to look at cytokinins and to meristems. You can micropropagation in C is axillary shoot development. That's grace to the counteracting effect of cytokinins, counteracting apical dominance. But you also have a new formation of meristems, advantageous shoot regeneration. That's mainly applications in biotechnology. Biotechnology, searching for new varieties by mutagenesis, CRISPR-Cas transformation, and so on. The way to look to it I think in a modern way is not just to add cytokinins, but to have a look what's happening with the cytokinins when you add it, and to regulate the homeostasis in vitro. Because when you measure the cytokinin level in the plant, you always find ranges as nanomolar ranges. In contrast, when you add cytokinins, it's mostly in micromolar concentrations, let's say 1,000 times more. This is like a enormous wave of cytokinins coming into the plant, and sometimes it's not prepared for this, and sometimes it's prepared and it wants to lower down all those cytokinins by all kinds of strategies. So when we have the input, it makes its own cytokinins, but you can also administer it. Then they can be interconversed. I will show you some examples later. They conjugated by sugars just to detoxify, just the same way as we do in the liver. Human liver also continuously detoxifies strange molecules entering in the body, and also degradation, of course, completely breakdown. So if this is a cytokinin 2IP, what can happen? It can be synthesized by the plant itself. It can also be administered. They can often the plant starts to interconverse by putting a ribose on it. It can also be converted into zeatin, for instance. Only difference is this OH group here at the side. So this is, let's say, interconversion. Nothing happens really to the activity, because you know zeatin is also a very active cytokinin. It can go both ways, in fact. But then it starts with the glucosylation. The conjugation with glucose here in this position, the O-glucoside, is still reversible. The glucose can get off and on. It's different when you have the 9-glucose here, when the glucose is attached to this nitrogen. It's more stable. This bond is much more difficult to break. And it can be broken down very slowly, and it can have an effect too on the routing processes, for instance. It can also be completely degraded. Okay, but maybe first we go to the school again, and we look to that cytokinin molecules just to explain a bit the abbreviations that I will use. So this is the 2IP, as you know. You see the changing of the OH to zeatin, ribose attached. I call this ZR by zeatin riboside. Also benzyladenine, you know, benzylaminopurine, BIP, BA. We use both abbreviations. I will use BA now. Here we have hydroxy groups can be attached as the metatopolin, but yes, also ortho in this position. Metatopolin can be here. And the paratopolin I don't show because it's not too much activity and it's not used. So orthometapara, but okay. It can again be ribose, MTR, I call it, and so on. These molecules, the metatopolins, I had the honor to test them for the very first time on tissue culture plants. And I think it was in 1993 or 4. I got them from Professor Mirek Strnad. At that time, he was not professor, but okay, we were working in the same lab in Antwerp then. He gave me some special molecules that they discovered in plants, benzyladenine derivatives in plants. And poplar, for instance, they found benzyladenine orthotopolin, metatopolin, orthotopolin riboside. It was a very surprise because before they thought benzyladenine was just synthesized, synthetic. And that was a start of testing a lot of differences between BA and MT. I must say, when I did my PhD, I also published some papers about metatopolin. It took many years before the tissue culture company embraced the topolins. That's always the case. New tools appear, and then oh, it takes so long time before the people find it. So that's also good maybe for this lecture, that it makes the time shorter to apply it if you know earlier. But the Metatopolins is not really new anymore. Many articles are appearing about it. Almost every week there's one. We also have other cytokinins, as you know, the diazuron. It's a completely other group. We will also touch this group later on. Completely other molecule structure. Yeah. But of course, this is not the ending, and there's many cytokinins were developed and are developed, mainly in Czech Republic, in the Institute of Experimental Botany. They dedicated in cytokine research, let's say. And they are the wizards that make all kinds of combinations, and they do it in a very systematically way. Let's say this is the benzyl, maybe the topolin or benzyladenine backbone, and they just call the groups systematically 2, 3, 4, 5, 6, and so on, and they just start to combine, adding everything they can from their chemical toolbox: chlorine, fluorine, methyl groups, O-methyl group, and so on. And they tested in particular bioassays. That's the Carlos bioassay and isolated wheat leaf bioassay and amaranthus bioassay, which is not really tissue culture, but it allows them for a quick screening. And I had the luck that often they send me like the best compounds to my lab to test it on a variety of plants to see if it's something interesting there. You could maybe wonder, why is this all this effort for the cytokinin research? Is it so important business, this tissue culture? Maybe it is for all of us, but not for the rest of the world, probably. The main reason for this injection of a lot of money was in the beginning, it was the cancer research, because they discovered that the benzyladenine, and derivatives, they had anti-cancer activity. That's why a lot of money was injected in this institute, in this research. So it's a bit spin-off that we can use as tissue culture industry, these new compounds. At the end, it was not so easy to prove really this anti-cancer activity in the last stages of the clinical research. It's also very expensive. And I think they abandoned a little bit this pathway to anti-cancer treatments. But anyhow, now we have all those nice cytokinins in our toolbox. Actually, cytokinins are healthy, so don't mind if you eat an apple and then you eat a lot of cytokinins or a lemon or so on. It's all there. Okay, so maybe I can share some small results to show the importance of the difference between those two benzyladenine and the metatopolin. Do you know this plant? It's Pathiphyllum. Very early in my career, I got this problem from the companies. They propagated the plant with benzyladenine. And when they wanted to acclimatize the plant under the best conditions, they saw it took so long to get roots. After 9 weeks, the plant didn't get any roots and it died. And what happened, I analyzed this as the benzyladenine. The plant was glucolized by glucose on the end position and accumulated in enormous quantities in the callus. And there it stayed, and it inhibited the rooting. Slowly it was broken down to BA again. And you know, cytokinins, they inhibit the rooting. And this plant could not manage to develop roots because of this enormous amount of glucosides. When you would remove the callus, then it would root again. So it was really this blocking with cytokinins. Then I got this molecule from the checks. I tried it, and I saw it already in the tissue culture recipient, there was already roots. So I could imagine that rooting would not be an issue for the cytokinin. And indeed, if you remove the roots, put it in the acclimatization greenhouse, very fast. After 1 week, you get roots, sorry, and a healthy, nice plant that is growing. Here, the metabolism was completely different. The sugar was not bound on the end side here, but the ribose came here, which is a reversible molecule, and the glucose was mainly put here on this side, reversible O-glucoside, which could be broken down very fast and released very fast. It was everywhere in the plant, but don't mind, it was broken down. So this is a story about this cytokinin metabolism difference. And we see now in many plants, this is the main reason why topolins give better quality of the plants and don't inhibit rooting, in contrast with benzyladenine. Voila. Good. Here I will also show you some experiment with pears that we did in temporary immersion bioreactors. You know, maybe I can first show this system. It's also nice to have a quick look here. This is it's a city system. Two recipients are stacked, one for the plants, one for the medium, and the plants grow here on this in the culture recipient for the plants. Every 6 hours, the air pressure is put here, and the liquid medium is pushed upwards and is immersing temporarily the plants. There's a backflow by gravity, spontaneous, if the pressure is released, and the medium goes back to the medium recipients. And it goes every 6 hours, the cycle. And moreover, there's also a headspace flush. Here, there's also a flush of new air pressure, which is going through this and bubbling here and released by this way. But in the meantime, the headspace is completely flushed, ethylene is washed away, and so on. The plants like this a lot because they have a lot of oxygen, a lot of food, completely immersed by the medium, and also the ethylene and other volatiles are always flushed away. So we use the system for pear. It's just to show this small, table to show difference between the semi-solid agar, medium, and cities, but also between those benzyladenine, MTR, MEMTR. MEMTR is the metatopolin riboside. We don't go into much detail here. I just want to show you this extremely high number of shoots with cities and MEMTR. So you see, and it's not very clear in semi-solid. So in every culture system, you can find some optimal cytokinin. It was very clear that in the city system, that MEMTR gave a lot of shoots, 14.5 shoots. But it's not only about shoots, of course, also about quality. But even when the shoots are small, and you can still build it in a system, and you can say, for instance, okay, let's do first multiplication stages is MEMTR, because it's a lot of small shoots, a lot of leaf area, and then you go to, for instance, MTR here, in the last step, to produce longer shoots with even more leaf area. So you can shift in the multiplication phase and then in the final phase to another cytokinin and produce the shoots in an effective way. But just to show the differences in quality and according to the system, of course. This is the plants that we produced. You see a lot of shoots, but maybe not the best quality, but a lot of shoots. And then you can go to this system, this cytokinin, to in the last phase to produce nice shoots, non-hyperhydric with a good leaf area and so on. This is cities, this is semi-solid. You see a difference in quality. You know. This is also a tool, this temporary immersion bioreactor, of course. So we could conclude here that benzyladenine was as efficient as MTR on agar medium, but hyperhydric shoots, MTR very high shoot proliferation in the cities, and MTR the best quality. That's just an example. Another interesting effect of these topolines was that chimeras are more stable in tissue culture with these topolines. And this is petunia, variegated petunia with the green core, L3 layer, and L1, L2 are white. No, I will not go into detail about chimeras because chimeras is an extra lecture, let's say, about chimeras. But this is difficult to reproduce because the layers of this petunia always try to get to mix a bit when you use the classical cytokinins. You get white shoots, green shoots, or even sectorial chimeras, half green, half white. But the customer wants this nice ornamental petunia. And here you see an experiment that shows the difference. We see here a raising concentration, doubling concentration of cytokinins, and there's two of them, benzyladenine and MTR. And you can clearly see this MTR preserves chimera structures even in very high concentrations, whereas benzyladenine also in the very low concentrations already you see white shoots, green shoots, completely useless in this micropropagation system because you get all kinds of mixtures. So the company who asked me this to solve this problem was very happy, of course. Just by changing the cytokinin, they could reproduce this petunia, make thousands and 10,000s, while the competitor was not able to do it because he did not have the knowledge, the simple knowledge about this topoline. Yeah, so this is BA, and this is the only difference is just this methyl group added to it. And also the changing of the metabolism, of course, too. Another nice thing we found about topolines that was the anti-senescing effects. You see here the roses in vitro. This was a bit, I found this a bit by serendipity. You can call it serendipity. It can also be like a bit laziness because I didn't clean up the old cultures after my experiment. So I tested all those topolines in the rose culture. And then, yeah, some old stocks remained there many weeks. And when I was cleaning everything, I had a remarkable observation that when you add benzyladenine to the medium with very early senescence, the plant just died after 2 months or 3 months, I don't remember anymore. Whereas that when you use MTR, that plants kept very nice freshly. So it was anti-senescence effect of MTR, metatopoline. You see, methyl, methoxy, metatopoline. That's a good message. The bad message, it's still a little bit expensive, more expensive than BA. So yeah, maybe if you don't need to switch to MTR, you don't have to do it, of course, because the price is maybe 20 times more expensive. But if you want good quality and long-lasting cultures, it's interesting. So this is the anti-senescence effect. No, the cytokinins were coming the next years because of course the biochemists, they didn't have a rest, and the ones they sent to me then, that was the fluorine compounds. Fluorine is a special molecule. It doesn't exist in nature, and they make it, the chemists like to add it to the molecules to make them more stable. And this is how it looked like. It was the metatopolin structure, but they changed the OH in fluorine here, and was also ribose attached to the molecule, you see. And yeah, this was special. Now you see, how can we change the homeostasis in the plant by changing topolin maybe, but also to use the fluorine, because the plant has many difficulties to break this down. Medicines and pesticides also contain this fluorine. 20% of all commercialized pharmaceuticals contain it. For instance, Lipitor and Prozac. Lipitor is anti-cholesterol and Prozac antidepressivum. It's great, thanks to the fluorine, it's more stable in the body and it's not broken down so fast as other molecules. It could also be a problem, of course, for the waste, because also it in the water, in the town wastewater, they found still traces of Lipitor and Prozac because it's so stable. And the people use it, go to the toilet, and the Lipitor is deposed there, and yeah, you see. But okay, we try to use these molecules also in the plants, because it extends the half-life and it enhances binding interactions. Very impressive results we got with Phalaenopsis. Phalaenopsis, we added 3 molecules, benzyladenine, but also the fluorine and the fluorine riboside topolin variant, you see here. So, and we, well, I know from previous experiments that orchids and also Phalaenopsis, they like this, they react differently according to the filter that is put in the lid of the pot. If it's filtered, then there is aeration. If no filter, completely closed, really completely, then they react different. Most plants, they don't like this complete closure and they like the filter, but not Phalaenopsis. That's why I combined both recipients with the 3 cytokinins. And you see, BA gives a nice plant, 2, 3, 4 shoots, as most people are used to that do the Phalaenopsis micropropagation. The fluorine MT, yes, strange enough, we got roots here, and which is also nice with the filter, that with FMTR, the number of shoots increased a lot. So people who do the Phalaenopsis know it's a slow plant in tissue culture. Let's say every 3 months, you have maybe 4 new shoots, so it's very slow to build up stock. Okay, but when you see this FMTR, it's nice. You have then every, let's say every 10 weeks, you can multiply by factor 10 already, and the competitor who's doing this, using this BA, is far behind you when you use the FMTR. It also changes when you use the closed container. Without filter, you see this also rooting is completely different effect here. FMT is producing more shoots, but have a look to this bunch of shoots when you use closed container and FMTR. It was maybe 30 shoots here. Too much. You should lower the concentration to have like a decent propagation. So it means that you can shorten the time to make stock from one plant in a very significant way. Of course, this plant is not so very nice to acclimatize, but then there's no problem. Then you simply go to FMT in the last culture, or even BA. You can shift from the very fast producing subcultures to the cultures which give normal plant again at the last phase. So this really went as a shock to the Phalaenopsis tissue culture world. Of course, the companies will never tell you what they use in their medium, but I know from inside information that really they use it a lot. This was also published, this article, so it's not really a big secret now. Yeah, so maybe it can also be used for other orchids. Why not? There's so many orchids and we cannot test every orchid, of course. This is all about micropropagation. Let's go now to the adventitious shoots. Adventitious shoots, regeneration, that's new formation of meristems from cells that are not used to do it or not destined to do it. We use it for chromosome doubling. It's a nice technology. It's still a technology which is used because chromosome doubling is not a GMO. Making not GMO, you can reproduce directly and put it on the market. We use for mutation induction. It can be irradiation. It can also be CRISPR-Cas, of course. Genetic transformation, gene editing, yes, mutation, targeted mutation. They all rely on protocols to regenerate a single cell to a new plant. For many plants, we must say this is still not feasible. We're still struggling, and every tool is welcome. Tissue culture, as a lot of you know, it's also trial and error. But okay, if you understand a little bit more of the plant, you can reduce the number of trials and make more educated guesses how to tackle the problem. But this adventitious shoot is still a bottleneck for all those nice applications. And I think also gene editing raised the demand for protocols for adventitious shoot regeneration. Yeah. For this, I would like to tell you about very interesting tools that we have nowadays. Not only recently we got them, very recently, of only a few years. Then you have to have a look to the degradation of cytokinins. Normally they are degraded by cytokine oxidase dehydrogenase. It's a long name. Let's call it cytokine oxidation. It's the fate of the endogenous cytokinins like isopentenyl adenine 2IP to be broken down to adenine and methyl 2-butanol, and then they just maybe recycled. Anyhow, not active anymore. Adenine has maybe slight cytokinin effects, but it's not very active. The cytokine oxidation, we can also illustrate by, I made a symbol from this isopentenyl adenine. Yes, the cytokine X. This is the red circle. It looks like a bit, I added an eye just to have like make it like a monster or so. And it breaks down that molecule and also show it like this. This is the cytokine oxidation. It takes the 2IP, breaks it, releases it, and again you see the two fragments, and it takes again, and the circle goes round and around. When you add 2IP to the medium, that's what this monster is doing. It's just taking it, eating it, breaking it continuously in a hopeless effort of the plant to reduce the concentration from the micromolar to the nanomolar concentration. So how can we inhibit this? This was a nice exercise. Of course, I must give the honor to not only to not to myself, but to the guys who made these new molecules. So they try to make molecules that inhibit this monster. and they produced like inhibitors of cytokine oxidation. They inhibited how you see this is the monster. It also can bind to the cytokine oxidation enzyme, but it stays there. It blocks. So yeah, the enzyme cannot do anything anymore because its active site is blocked by the cytokine oxidation inhibitor, who has the same form, even better form to attach here, and it cannot be released anymore, or very difficult to release it. And yes, what is the nature of these inhibitors? It's already a long journey, chemical journey of, I must say, the most of the Czech Republic people of the Institute of the Experimental Botany in Czech Republic. A lot of them worked on this. So we had like 2 IP analogs that they made. That was published in 2008, but they were difficult to produce, to synthesize. They didn't get the bench of the experimental tissue culture companies and so on. They didn't use it, but it was a good trial. Then we get also 2008. They tried to change the metatopolin riboside, the one we just saw, just by adding chlorine here or fluorine, and they introduced inside fluor, inside chlor and inside fluor. They at that time they already introduced like way to measure the activity of those cytokinin inhibition, cytokinin oxidation inhibition, the IC50, the concentration to bind 50% of the enzyme. So the lower the number is, the more active. 1.9 micromolar was good to bind 50% of all those cytokine oxidation enzyme. Fluorine was even better, 1 micromolar. This was published, taken up by some people, and they got some nice results, but we wait for other new, most stronger molecules, in fact. And they came 8 years later already, and then they made efforts to change the TDazuron molecule to add some extra groups, fluorine and so on. You see, the IC50 was not improved a lot. 3.9, 5.5. If you compare with the 1.9 and 2 of the previous molecules, it was not an, it doesn't seem to be very interesting, but the synthesis actually was more easier and the molecules were cheaper. And they saw that it was a very good strategy to develop these molecules, and now they tried more modern tools. They designed in silico on the computer. They designed the best inhibitors. You have active sites, and they calculated the structure of the protein, so what was the best group to attach to it to have a best binding. And then from this research, Jaroslav Nisler, he was the colleague who did this research. He started from diphenylurea compounds and changed them to put bromides or fluorine, even 3 of them, and he developed very powerful molecules. And that's the ones that I will show you some results of. You see the IC50 is very low. It's 30 nanomolar already. This one is also very good, but this really is a fantastic molecule because it's only 1.030 micromolar, inhibits already 50%. If you double concentration, almost everything is inhibited. So I share you some results with this molecule. It can inhibit the breakdown of 2IP and zeatin. Okay. I was just trying to. Yeah, here it is. This is the molecule of the 3 fluorines. It's actually this number 1, with the 0. The most active one. It's nice to see here, if you can still concentrate, because I know it's a lot of information now, but this is very nice. If you combine 2IP here, that's the cytokinin, with that compound and raising concentrations 0.01, 0.1, and 1 here, and the same concentrations of 2IP here, then let's have a look what happens if you add nothing. This is tobacco leaf disc, all of them. The leaf disc doesn't do anything when there's nothing in the medium. It's MS medium, it's sugar, and so on. Yeah, if you add cytokinin, 0.01, nothing happens. 0.1, okay, shoot induction happened, and we get a lot of shoots. So this is a normal way to have a look to it. What happens if you add the compound here? The compound without cytokinins, nothing happens, nothing. So they don't have any cytokinin effector compounds. They just hunt the cytokine oxidase enzyme. And when you add a little bit of 2IP, normally the plant brings it down, nothing happens. But with a small amount of this inhibitor, you see already some differences. A little bit more, 0.1 micromolar, and yes, you have shoots. So the 2IP, which was added here and broken down, was protected more or less by the inhibitor, because the inhibitor bound on the red monster there. You see, it was completely inhibited, and the 2AP could do his work and give shoots. This is the important place of this table. When you add more cytokinins, you get shoots, even more. You see this. The protection is it's not so clear because you add already a lot of cytokinins. So here it's like overshooting 2IP, and but this is the important place where it happens. That shows that very low amounts of cytokinins can induce shoots if you protect them from degradation by also a very low amount of this inhibitor of the oxidation enzyme. I will show you some more results of this. For instance, Plectranthus is, maybe you know it in Indonesia, it's edible tuber from the Lamiaceae family. And it's a bit of the same experiment. This compound we called at that time GN1 because it was made by Jaroslav Nisler. You see, if we go up, sorry, Nisler. He sent me these molecules. Sorry. I have to go down now. Maybe it's not a good idea to switch too much. Yeah, here. So when only GN1 is added, let's say, nothing happens, or maybe it's even some bit negative. 2IP gives a few shoots, but when it's protected by the GN, you get more shoots. So I show you different combinations now, where the combination of 2IP with GN gives almost more shoots. For instance, for the lobelia, it's a flower, ornamental pot flower or bedding flower. You see, control, nothing happens. 2IP, small number of shoots, a lot of colors. But when you start combining with the GNs, you get a lot of shoots. We got GN1, GN2, 3, 4, 5, and it continues on. Now we're on number 58, let's say. Too much, of course. So there was already some selection, but we discovered very strong ones. Like number 4, very strong. It seems that you have to find a good combination between the GN, the cytokinin, and the plant to have like a lot of shoots. At tissue culture, people like this to find the magic combination, because you can keep, also companies, they can keep it secret and so on. But it gives new tools to experiment with a lot of species. For instance, lavandula, you see, it's 2IP doesn't give any shoots, just colors on the leaves. The GN1 was not improvement, but here it illustrates that you have to find the key. That number 4 and 2IP was the key to find regeneration. this is not just like fun for the lab to find this for the researcher, but it means you can do mutation induction of lavandula, you can do CRISPR-Cas with lavandula, doubling chromosomes, everything much easier when you find this key for regeneration. So it has a big implications of the to find this combination. So we should be forever thankful to the Czechs there that they could make this molecule. And I can share the you can find Jaroslav Nisser on just by Googling him, and he's open for cooperation because they filed the patents, but they don't want to keep it secret. They want to that everybody uses this molecule just to share the knowledge and to publish about it. That's the best strategy to have to become, let's say, famous forever for Jaroslav Nisser, just to share it and not keep it in his own lab. Another example. This is the carnivorous plant Drosera rotunifolia. Maybe you have it also in Indonesia, I don't know, but it's catch the flies by sticky drops on the trichomes. And here you see something else. Even when you don't put any cytokinins in the medium, it can regenerate some shoots. It means it makes also some own cytokinins in the leaves. And when you put a GN, it enforces the own endogenous cytokinins. Of course, when you add to IP, you get shoots and you get even more shoots, but when you combine it. But it shows that sometimes you see that also endogenous cytokinins allow to regenerate shoots without adding any other ones, and that you can stimulate this by adding this GN1. Oh, yeah, my students do a lot of combinations. Let's see, here's potato, for instance. They combine no cytokinin, 2IP, 2IPR, zeatin, BA, all of them. Although we know BA-MTR is not so sensitive for this oxidation. But you see, there's no results with these molecules. But if you combine 2IPR, you see here, combine it with the number GN2, another GN of the family, then we could have shoots. So very easy to get shoots here if you find the right combination. So I don't say up to now there's no regeneration protocol for potato, but it can help anyhow to increase the number of shoots if you do transformation experiments or mutagenesis and so on. This is all about this molecule. And then, so we can conclude here, there's a number of molecules with really cytokinin activity, and there's very small cytokine oxidation inhibition because they all fit in this. It's the same, like the same cassette here or here, it's the same. But if they attach here to the receptor, then they give shoots and the cytokinins, but they can also attach to the inhibitor, they are broken down. But some molecules are, let's say, inhibitors, more inhibitor, and more others are more cytokinin lovers. So T-diazuron, CPPU, they're more in this level. So you see their cytokinin activity, but they also have a small cytokinin oxidation inhibition. Long time they thought that this was the only way that T-diazuron worked, but it's not true. It's also cytokinin effect here. If you go down, we see the insights that we just showed. The first, one of the first generation inhibitors, but the new compounds, they are really here. No or almost no cytokinin activity and very strong cytokinin oxidation inhibition. Yes, that's the one we are focusing on. A last slide about this oxidation inhibitors. It's also good for somatic embryogenesis. Not in all plants, of course, because most plants, they need 2,4-D or other auxins from the same family to start that somatic embryogenesis, callus, and so on. But there's a difference in coffee. In coffee, it's different. In coffee, you can induce somatic embryos by cytokinins. See here? And we could also stimulate the effect of 2AP, which you can use for somatic embryogenesis, with this oxidation inhibitors. We could normally for coffee, you have to make its indirect somatic embryogenesis. First, you have to make the callus, then subculture the callus from particular size, and yes, so the embryogenic callus lines, and then after many months you can start producing somatic embryos. But here we could see direct somatic embryogenesis formation on the leaf disc. So very fast, after only a few weeks, you could produce somatic embryo on the coffee. See, so that's also a nice application of this inhibition effect of these molecules. This the embryos, and then a nice plant, just like a seedling of coffee. Nowadays, they use somatic embryogenesis in coffee, not really for the production to go to the field, but to make mother plants. You can make easily a few thousands of mother plants with this technology. And because the plants are very juvenile, you can use them for making cuttings in an industrial way. And that's nowadays they do for the coffee. But this technology, this molecule can help to make in a very fast way somatic embryos from new varieties. So it can also have impact on a cup of coffee you will drink in the future. This is the molecule. Completely other tools. I hope you're not saturated at this point. Maybe you can have a break somewhere. Yeah, so maybe Weirda, if it's time for a break, just call, and then we have a short break for some questions. But maybe I can just finish this small chapter about enhancers of advantageous shoots. This is a picture of also spathiphyllum, and you see also by serendipity, I must say, we discovered that when you add imazalil, which is a fungicide which is still used in horticulture, for instance, they spray it on grapefruit, oranges, and so on, to prevent molding. This molecule, and if you combine it in the medium of with benzyl adenine, you get a lot of shoots. This is the normal number of shoots you expect from a decent culture of spathiphyllum, 3-4 shoots. But when you combine with imazalil, you have hundreds of them. It's incredible. What is happening here? And the imazalil, in fact, this is the molecule. It has a special ring here with 2 nitrogens, and especially here there's a free electron on that nitrogen. Normally, it's active in the steroid biosynthesis of the fungi. It inhibits that the pathway of the sterols, and that's why the fungi don't grow and your oranges are protected. They bind on P450 enzymes. That's a whole group of enzymes active in the whole living world, let's say, or in mosses, in higher plants, in humans, in fish, everywhere. In the metabolism, P450 enzymes are active, but particularly this imazalil binds on the one of the fungi. But of course, as you know, the diversity is so enormous in the life that they could also bind on other molecules, on other enzyme P450 enzymes. For instance, in the plant, the imazalil is binding and it is inhibiting. That's what we found, brasinolide, the brasinosteroid hormones. And here, for some reason, they gives a lot of shoots. This 2 genes, DWARF4 and CPD genes, that are producing an enzyme, a P450 enzyme, that is inhibited by the imazalil. Yes, okay, we could have a whole lecture about this brasinolide 2, but to be in short, what is it doing? it's responsible mainly, the brasinosteroid in the plant, for the dark germination. If it's, this is like a Arabidopsis grown in the zone in darkness, is germinated, looking for light, making a very long hypocotyl just to reach the sun. With this, for this elongation, brasinosteroid is responsible for the elongation of the cells. If you add the IMAS allele, which is inhibiting the production of this steroid hormone, the plant stays short and it thinks everything is okay, it's in the light, but in fact it's in the darkness. It grows like a normal plant, opens the cotyledons, waiting for the sun, which will never come, of course, if it's in the soil. If you, yeah. So, but just to show that this is the control plant. If you add the IMA, you get the small plants. If you put the GA, it gets a little bit bigger. If you add the IMAS allele and the synthetic brasinosteroid to recover the plant, oh, it's not a nice plant. It's just crawling over the medium, losing any sense of gravitropy. If you combine the IMAS allele plus the 2 GA3 and brasinolide, you get some restoration of the plants, but still not very normal. It's not straight growing upwards, but just crawling on the soil. It has no gravitropy. So still other things are happening in the plant with the IMA, which we cannot recover with these molecules. But you see, that's how you can find the way that everything goes. But anyhow, the effects that we saw here with the spatifilum, I go back, probably it was caused by brasinosteroid inhibition. Yeah. Of course, it's not so nice for the producer of this IMAS allele that it has side effects on the plant. Not on every plant, probably, but on some particular plants, you can use this molecule for regeneration. And at that time, we used this effect for doubling chromosomes. So we put spatifilum shoots in a medium with colchicine, or it was orizaline, I think. We got a lot of shoots, and a number of them were just doubled in chromosome number. That's how we made doubled chromosome spatifilum. I will continue back. Yes, maybe it's good to have a break now. If some questions, I will stop sharing now. If it's okay for
yes.
you.
Prof. Stefan, we have questions in the chat box of Zoom.
Okay.
I will read.
Okay.
I will read it for you. First one is from Elok. She's from Indonesian Research Agency. I work with on the in vitro propagation of some dipterocarps, but unfortunately the explant failed to give me response after 5 months. I use BA for the cytokinin, but what should you suggest for this problem since you also work with woody species? It's dipterocarps.
Pterocarpus.
Yes, dipterocarpus.
Oh yeah, but I didn't work on that crop. But for the shoot induction, for the advantageous shoot induction, that's the question.
Yeah, I guess so. I guess so.
I would. Yes, I would use these fluorine compounds. They can already be bought, and that's in the company in the Czech. The Czechs have also a spin-off company where they sell those new compounds. It's Alchemim, alchemim.cz. Maybe I can also put it in the chat. Yeah, and in the chat. Yeah, why not? To write it, the right correct name, CZ from Czech Republic, is sent to everyone. Yeah. How can I send it now? Did you receive it? Yeah, okay. Oh, this is the one. You can also, those GNs, the compounds, are not yet commercially available because we're just publishing results now. But you can write a letter to him and ask for some small amounts to test it, and he will probably bargain with you. You say, okay, I send you, but maybe you can put me co-author if you find something. If you don't want, just pay it. Just try. Yes, I can also copy his email address, of course, if you want. One moment. It's just a Gmail. I will just copy it. Yeah. So that's his email. So I would really try this combinations 2IP, zeatin with a GN, some GN, some of those compounds of him. You could also, yeah. But I will show you more tools. Maybe you can have more inspiration by the next step of the lecture. Yeah.
Miss Elok, are you still with us? No, I cannot find her. I think she's not here anymore. But we will forward the information to her. And there is another questions from Dini, Miss Dini from ITS University. The question is, I'm Dini from ITS Surabaya. Very interesting material regarding growth regulators. I want to ask what exactly we should pay attention to in choosing the type of growth regulator to be used in plant in vitro culture. Do plant species also greatly influence the response to different growth regulators?
Yes, of course. Yeah. Every plant reacts differently. You see in nature, some plants grow very high, some branch, some go off the ground. It's all about plant growth in a regulator, receptors everywhere, and they can be different in this every cell. So it's really about testing a lot of them. But anyhow, we know already where to find. Some molecules are very active in a lot of species. Yeah. But it's like cytokinins. You also have to know a bit that, for instance, the Ericacea family, they don't like benzyladenine, for instance. The general rules you have to apply. And we know some of the best inhibitors, they work on a lot of species. So you don't have to do too much screening.
Yeah. If anyone have any question and want to ask directly to Professor Stefaan, you can just open your microphone and ask it directly. Otherwise, I have questions, Professor Stefaan, regarding metatopolin that you explained, that it has a stronger effect compared to IP and benzyladenine, as you explained, to induce shoot formation. Does metatopolin bind to the same cytokinin receptor as benzyladenine, for example, and does it's activating the same gene cascade, the cytokinin responsive gene as benzyladenine, or it's a different pathway?
Yeah, that's a good question. We don't know exactly, but the difference is more about the metabolism. So it's not glucolized like the benzyladenine and released very slowly as glucose. But I think we see the same effects of shoot inducing. But this about receptors, we don't know exactly. I think it's the same receptor, but I'm not sure. Of course, in science, you can never say, I think. You have to prove it. Huh? Yeah. They never really proved this in all those species. They even don't know what the exact receptor in, for instance, platyphyllum is. So there's a lot of work to do. Yes, there are many receptors, of course, in every plant, and which one is more responsible for shoot regeneration in vitro? Yeah,
Okay.
Still a lot of work to do for young researchers.
I see. Does it ever tested in Arabidopsis, let's say, this metatopolin?
Yeah, I think so. Yeah, it's also there. Yeah.
I mean, like,
but Arabidopsis is a bit a difficult plant in tissue culture. It's a very nice model if you use seeds and all kinds of mutants, but in tissue culture, it's behaving very weird. It's not the ideal plant, let's say. Yeah.
Okay.
Yeah. But I'm in my career, I'm mainly focused on other plants, on really crop plants, crops. Yeah.
I see. Yeah.
Actually, I'm still trying, and not still trying to do cacao. Cacao is very hard to attack tissue culture of it. Okay, somatic embryo is okay, but then the next steps is very difficult. Yeah. So until now, we still have to continue the research with cacao and use the new tools on it.
Yeah. Okay. Thank you, Prof. Stefaan. Is there any questions from the participants for Prof. Stefaan so far? Okay. There is
I see
one
something about
question.
contamination too, but I will continue. We'll see. We come to contamination too. So we see some.
Okay.
Okay.
Yeah, there is a questions from Arif Yahya. Hello, professors. We know that contamination is great. Okay, so you will ask this later on, or should?
Yes, but it will be more about virus contamination. But about bacteria, yeah, that's something else.
Yeah.
It's a complete other lecture about maybe next time contamination.
Okay.
But it's also very interesting to see what you can discover in the plant. Sometimes it can be a complete collection of an ecosystem of bacteria in the plant, which you don't know. You think it's sterile. You don't see anything, and then when you start to investigate, you find the whole ecosystem there. Very interesting. But no, let's go to the tools. I will continue with my lecture. Yes, I will share, and then at the end we can have more time for questions.
Okay. Maybe one question about cytokinin from Professor Wulan Manuhara from UNER. Do you have experience about using two different cytokinin together to induce shoot formation? Is there a good induction or a bad one? Like if you combine.
Yes, it's also good to combine cytokinins. That's also an interesting field. Of course, it increases a lot. Number of combinations is more increased, but I know there's protocols that combining benzyladenine with topolins, and they lower everything. It's very effective. Or they shift BA, MT, BA, MT. It's a bit to reduce the costs because MT cost 10 times more than BA. See, not if you calculate per liter, it's not too much, but okay, every lab has to reduce costs, of course. Yes, there's also an endless variety of combination of cytokinins. Yeah, but not too much publications about this. The companies do it, I know, I'm sure, but the companies, they don't publish their best medium. No.
Yeah,
that's a pity, but yeah.
Yeah, or maybe you can use the whole cocktail of GN and then two cytokinin.
Yeah, but GNs, I don't know.
GNs
will not work very much on the aromatic cytokinins because they are all more or less protected for this cytokine oxidation. But yes, you never know. You never know if something is well improved. Okay, I go to share my next slides because I still have a lot to tell you, of course.
Okay.
It's really a lot. So that was the last slide. I hope you can see it. Yeah, this was the last one. No, I go to the next slide. Yes, and that's about nano technology, let's say in vitro culture. There's different, of course, nano covers a lot of interesting technology, but here, what did we do? In the lab, we make small particles of calcium carbonate by mixing these molecules, two of them, these chemicals. You get a chemical reaction and get calcium carbonate and salt, sodium chloride, but this calcium carbonate with some particular technology, you can make nice spheres, round spheres of very small, 1 micromolar diameter. You add, for instance, auxin molecules. You put 1 hour in the shaker, and they're attached in the nanopores of this calcium carbonate particles. These particles, they are like micro, but they have nanopores, which can, where the cytokinins can enter and be fixed like a battery. Of course, it's very small to handle, and you have to aggregate them. Yeah, aggregate mean like big structures of millions of those small micro spheres. Here you see it in reality. Maybe I can make it a bit bigger for you. If you can see it. Yes, this is the microparticles. The size is 1 micrometer is the size of a bacteria. These particles. And then we make the milliparticles, which is like size of 1 millimeter, let's say. You see all the millions, billions of small microparticles. Every particle has absorbed a plant hormone. For instance, IBA or cytokinin or GI. Some molecule can, no matter what molecule. And this is shown here. Is one, you can take it as 1 millimeter. 1.2 maybe. This is the size. You can take it and manipulate by small forceps, and it's the nice thing is that it is slowly releasing the IBA in the medium. You see, if you put like a leaf disc. Oh no, my mouse has disappeared. Sorry. If you, this is a small particle. I put it here, and this is the leaf disc. And this is the side from above, and this is the side from this side view. You see this particle releasing the blue molecule more and more and more, and it's reaching the leaf. And then you see some particular point, the shoot induction. See it here. Of course, the leaf also expands. It covers even the particle. It covers it. But no, wait, so you see, you can, it's completely different than putting the hormone in the medium, because then it's everywhere at every point the same concentration. The plant, they like gradients, so the increase of hormones, and then it's going away, it's diluted and going away. So this is technology that we are developing in our lab, just to see what happens at this moment. But you see here, in reality, this particle gives shoots. You can put it on the leaf, beside the leaf, somewhere you want, and it gives shoots here for tobacco. The leaf discs, so form shoots in response to the cytokinin that is slowly released by the microparticle. But of course, you see there's so many combinations you can make with these particles. And here you see an experiment of Saba. There's a lady in my lab. Saba, she did experiments. She shows here effects of the 2IP and the GN1, the inhibitor of oxidation, in the medium. You see, nothing in the medium. And the particle, there was nothing. It was control particle, empty particle. 2IP was not able at that concentration to induce shoots, but if you combine it in the medium with GN, as expected, we find here shoots. Yeah. But if you put the compounds in the particle, you see here, you see in the part, not in the medium, but in the particle, you see about the same thing. Nothing happens when there's nothing, and it's just control too. But if you put GN in the particle, the leaf disc is like more greenish, yeah, but no shoots. 2IP in the particle, nothing happens. But if you combine both, a particle with a GN and one with the 2IP, yes, you hit a lot of shoots here. So this also illustrates a bit, sorry, the effect of those inhibitor of the oxidation combined with 2AP, as previously shown, but also shows that if you put them in a particle, that you can have the same effect. And you can put them everywhere you want. You can put them aside. You see the effect of cytokinins on the leaf disc. This was only in these particles there were cytokinins, and they induce so much shoots here. It's so powerful. It's like a battery full of cytokinins that you put here by the forceps. Also auxins. They give shoots, a particle with auxins. You can put them everywhere, those particles around it. You can combine auxins. It's like a Lego toy. You can have a particle with GA, with gibberellic acid, a particle with GN, with auxin, cytokinin. You just put them on a certain place and see the effect. It's a very nice toy in the lab now, and many people use it. For instance, here's somebody with Arabidopsis. He does a germinating etiolated Arabidopsis, which is, this is seed is here, this is hypocotyl. They put on a medium, they made a composed picture, of course, because it was too big. You see, there's a particle with auxins, IBA. It's slowly releasing and it touching this hypocotyl and make a lot of roots. Here too, the zone is touching and make roots. You can see where everywhere the particle is, there's roots. So this is like a new toy in the lab. We don't know if we will have a breakthrough somewhere in some plant, but we can play with it and maybe also make some breakthroughs. So this is about this slow release. Yes, I also have to go to other compound, to another to leave this advantageous shoot regeneration and to go to the rooting, because also for rooting we have new compounds. This is the Melia volkensi, the first plant I showed, that tree from Kenya. Oh, when you add IBA to the medium, they get these plants. A lot of callus, a fat root, which is not functional, really not very good. The spongy roots, but we need this plant, of course. Not too much callus, roots attached to the stem, and functional after acclimatization. Here you see an experiment where we used cinnamic acids. It's also a compound that just illustrates a part of our auxin research, because we have also other compounds. This is the pear, a shoot of pear. When you add IBA, classical first choice to have rooting, you get too much callus here. There's no roots here, and then we found combinations of all kinds of effects. For instance, here we used white fluorescent light and compared with red LED light, and the combination of adding this transinamic acid, which is in fact lights, depending on the light, for which is, if it's cis or transinamic acid, it's like switching from one to the other side by light, active or passive form. So we add transinamic acid, but in fact it should become ciscinamic acid, which is active. If you put red light and combine it with this transinamic acid, then we get very nice rooting. Under cool white light, only 74%. Under the red light, probably it was shifted very nicely to the active form, and then we have this nice roots here. So also for the rooting, there's still room for improvement for a lot of plants, especially when you want to avoid this big calluses. What can you do with a plant with big callus and a root attached on it? There should be a connection between the root and the stem. Yep. Then we also have very powerful auxins, like a new generation of auxins we call it the compound auxins. That's like auxin attached to esters. They don't work actually as auxin, but they have to break down the ester bond and then they start working. Here you see, for instance, one of illustration of this compound in a plant. It's in poplar, I suppose. If you don't put anything in the medium, hormone-free, you get one root, yeah, and then you start 1 micromolar of this auxin compound auxin. You see what happens. It's taken up, probably broken, decomposed somewhere in the plant, and the leaf petiole is starting to root. Here too, you see, it's also epinasty showing ethylene effects. If you make even a higher concentration, 10 micromolar, the plant is transforming in a bunch of roots. The leaves are rooting, the petioles, the stem, everything is transformed in a monster made of roots. You see this? Incredible. So it's just too strong. Nobody can use this monster. So it's more matter of controlling this compound than adding it. If you add it, we are trying with short pulses of this compound, or very low concentrations, or a combination of it to root plants that before cannot be rooted. Yeah, the compound auxins, they're not yet on the market, but yeah, soon they will become. Yeah, I think, yeah. You see also here Helleborus effect, for instance, Helleborus on this compound. You see a lot of roots here, yeah, a lot of roots on the petiole. This is just the leaf. The leaves is rooting, bunches of roots with this particular new type of auxins that we have to learn to use because it's too strong. Okay, rooting, you have all the compounds, but also for acclimatization, there's new compounds. You know, acclimatization, often you fail because the plant is drying out before it's make roots. Even if you cover it plastic and so, the idea is to add abscisic acid to the plants to close the stomata. If the stomata are closed, you can imagine the plant will not lose water anymore, or less water, and it has more chance to survive. But the problem is abscisic acid is broken down very fast. After maybe 1 day is just finished. It's just broken down. That's why now there's some chemical analogs which are interesting, like the pyrabactins. Pyrabactin is, you see, this is abscisic acid to the right side, and here the big one is this pyrabactin. It contains bromide to stabilize it, you see. It's much more stable. It can close the stomata for a week or longer. So the plant is much better protected against this acclimatization effects, drying effect of acclimatization. I see nice roots here because the plant was protected. This is the same tree as we have before, the first slide. So this can make difference, of course, when you have nice roots. Then we can start planting the best trees of all those 3 million of individual seedlings. This work, and that I like very good, and like in this tissue culture work, you can make breakthroughs and have a lot of effects on the field. It's like a bottleneck you open, and then the people can start applying it, start planting trees because they have roots, start planting the best trees because you can multiply them finally. So that's a large impact of, let's say, a technology that is known by only few people in the world. Maybe I also have a nice, we had already, yeah, micropropagation, adventitious shoots, rooting, acclimatization. I want to also tell something about technology It's also relevant to eradicate virus. And here, for instance, I took one that maybe you also have in Indonesia, I suppose, the yam. Yam is, of course, basic food for in Nigeria and Cuba mainly, but I suppose you also have yam there in Indonesia. When we checked, it's produced largely starchy tubers for important food in the Caribbean and Africa. It's vegetatively propagated and it has virus inside. And I want to show you some new tools that we have for virus detection and virus, and then the tools to make the plant virus free. So we received some varieties from Cuba, where they're very big. Sometimes they grow very big, the yams. They make, this is the food they make, like from the flour they make like potato-like bowl. Yes, nice food. So what do we do with this plant that we received from the Cubans? For the virus detection, we pulled some plants and we extracted total RNA. And we send it for next generation sequencing. There's all the RNA we could send to the company, and it came back 40 million reads. Is this a new technology? No, but no, it's becoming cheap. What we call cheap, it's not like 5,000 euro anymore or $5,000. It's only $300 you pay, depending on the provider, sometimes 400 euro, and the price is still decreasing. So it's available now to, let's say, to send all RNA, and then we use bioinformatics, and the software detects all the viral sequences, and it gives you an overview of all the virus in the plant, even unknown viruses that nobody heard of. And we found a lot of virus in the yam. The next step, of course, is to do, you can also do cDNA PCR with this extract, and then you can check per clone by PCR, of course, yeah, because you have the names of the virus, we know the sequence, you can design some primers, and you can check if everything's there really. And also use this technology after the cleaning to check if the virus is gone from the plant. How can you do the virus elimination? Different ways. For instance, thermotherapy. Sorry for this broken word. So you heat the plant to almost 40 degrees in a special culture room, and the virus is the plants suffer from this a lot, but the virus much more, and it's excluded from the meristem. You do meristem culture, in vitro rescue of meristems, of course, and then you do it again, the test cDNA PCR to check if the virus is gone. Some results that we got from that yam, we get some African yams, we get some Cuban lambs, and you see how much virus is in the plant. Batna virus, yam mild mosaic virus, all those viruses we could detect. It's full of virus, and every small shoots you produce from that plant is also contains the viruses. So if you use this for tissue culture and you selling these plants, you just spread the virus everywhere in the country. As a tissue culture company, or you have the responsibility to distribute clean plants to the customer, to the farmer. The yield will be much higher because virus always take energy from the plant because they change the complete machinery of the cell to produce even more viruses. But how can we cannot sell this? Bela, for instance, it contains 7 viruses. So we could do the heat treatment in the heat therapy. 36 degrees was already a lot, because you see how much the plants suffer from it. They don't look very happy. But then we could, to take the small meristems by microscope, put them on petri dishes with charcoal, because it's so small and some of them regenerated. And in fact, it's effective. You should try it because you can say, oh, it will never work. The virus is everywhere. But no. When the heat therapy, it's very effective. And now we're also combining it with chemotherapy because now it's at the COVID time. We have a lot of antivirus compounds from the medicinal world. So we can also combine it with anti-mRNA virus medicines. And but okay, at this phase, we didn't use the chemical. We just used heat therapy, and we could have free the yams from the most of the viruses. For this also technology, if the merisstems don't grow, in this case, it grew, some of them died, but a number of them survived. But there's some technology called heterografting. And this is a very strange way to produce meristems. You can, for instance, do the heat therapy, take the small meristem, the dome even of the meristem, slice it. It's a very careful work. But if it's too small, it will not survive. And then comes in like an idea from it's mainly from a Japanese group. They take another plant, for instance, radish. They germinate in vitro. They take the root tip of the plant, which is completely not related with this tree, for instance, and it's like a nurse. The small root tip is like a nurse for the meristem. They are combined. Yes, the seedling is a bit naive creature. The roots don't expect this. They don't react by some reaction. No, they just think everything is okay. They continue growing and they feed the meristem. And when it's big enough, you can cut it off and put it on the normal medium to make a normal plant. There's also no virus in the radish, we think, because you could also think maybe we can add more virus by this way too. But okay, if it's like seedling of trustable seed company, it's not containing virus. And this way, by the meristem culture, you see, we could step by step eliminate viruses. Yes, don't show the rest because step by step we did it. And now we are at the point that we could deliver to the Cuban colleagues completely clean yam variety, except one of the viruses is this one, Batna virus. You know, Batna is, you see here the word DNA in it, DNA. It means it's integrated in the genome. So how can we cannot, yes, it's always there, of course. So, but it's not so serious effect, or you cannot see the effect too much of this virus. So it's not so harmful. We are very, very pleased to deliver already this completely clean yam, and we see what the effect on the growth and the yield in the later and months to come. So this can also have a big impact on the tissue culture industry, because more and more with this next generation sequencing technology, and it will become much cheaper, the governments will ask more and more virus-free plants. Even when you cross the border with plants, they will ask clean plants. If you can deliver them and the competitor cannot, then you are an advantage. And we have a big project on this here in Belgium with producers of cuttings and in vitro labs who are very aware of this, and just to continue with this cleaning and to checking and so on, to produce the best planting material for the farmers. So this is also a very important technology, I think, also worthwhile to mention in this lecture. So I don't know exactly the time now because I don't have a watch here, but maybe it's good to draw some conclusions and then we can have some more questions. Yeah, because I think it's in your mind must be completely saturated now because of all those new tools. But what can we have as a conclusion? maybe plant tissue culture is more than only cloning, of course, but even cloning, we have new tools. It's also interesting, but the actual evolution of targeted mutagenesis, CRISPR-Cas, let's say, requires single cell regeneration protocols. You need this advantageous shoot protocols to continue this fascinating technology of gene editing. Also, virus sanitation requires meristem culture protocols. It's also, yeah, we need it. So much research is needed, and also funding, of course. Yeah. New compounds that I showed you are becoming available step by step, and they push further the technical boundaries of the regeneration. It doesn't stop. We will always find new tools, new ways to do it. Just don't stick to the benzyladenine and TDazuron. There's much more tools, because yeah, it can make difference. Always nice to publish too, because there's always new compounds there. The journals accepted those articles. Yeah. Plant tissue culture, of course, relies more and more on educated guesses. You have to understand what's happening by analyzing in the plant. Yes, we also do this, analyzing the cytokinin profiles. I had no time in this lecture to go deeper into this, but it can help you to choose the right cytokinins, the right combinations. But of course, it's still a matter of trial and error. You have so many plants, so many varieties. Everybody has his own bottlenecks. But maybe, yes, this lecture can be like an eye-opener, and you can always mail me for more information or for connecting to my network. Maybe we can solve your problem. Of course, it's not a wonder formula which can solve everything at this moment, but step by step, we need to put more and more tools in our toolbox to advance in the science. So this is a number of people who helped me to realize all that research. You see, it's maybe a little bit older picture, but also the results sometimes are from that time. In the middle, you see here Mirni from Palu, which works on cacao, from Indonesia. Yeah, so we have many people from different countries in my lab still. Okay, so I stop sharing and I'm open for questions.
Thank you very much, Professor Stefaan, for the wonderful talk about pushing boundaries for tissue culture. We have two questions in the Zoom chat box. First one is from Ms. Dwi Kusuma Ayuni from Erlangga University. The question is nice topic. I just wonder whether some compound or the growth regulator used in your research affecting the metabolite composition and production in the callus or plant lab. So basically because we propagating a lot of medicinal plants. Let's say if you use BA or you use topolin for producing shoot or producing advantageous fruit, do you think it will change the metabolite composition in the cell or in the
yes, of course. Yeah. There's already so many. I could not say it 20 years ago, but there's so many articles now, hundreds and hundreds of them, that showing beneficial effects of topolins. It is really worthwhile to check it. Of course, I don't know every plant. So thousands of plants, also medical plants and pharmaceutical, and very interesting, of course, in Indonesia, where you have so much biodiversity, to use this and not to avoid harvesting in the wild. You have to domesticate it and to grow it in vitro and propagate it. So it's very important to check all those plants from strange families too. Yeah, really, I think they will make difference. Yes, of course, it's a bit they cost a little bit more, okay, but for research, no problem. But if they can make a breakthrough, give better quality, and also just be the difference between no and yes, then you have to apply it. Yeah.
Yeah, I think it will be very interesting to do metabolomic analysis in the plants produced using PA and then the standard auxin like IAA compared to using compound auxin and topolin to see whether.
Yes, it's nice research because you see very big effects. You have to do nice experiments, do some repetitions, and you can easily publish this because it's a new breakthrough in some crops. Yeah.
Okay, we have another questions from Ms. Nurmalasari from APRIN, Indonesian Research Agency. I think this is my first time learning new types of cytokinins, MTR and MEMTR. You mentioned about MEMTR function as a chimera stability in ornamental plants. How many generations that chimera character proven to be stable? Is the function as chimera stability can be used in crops as well? So
yes, in chimera crops. Yeah. So I didn't do 10, 20 generations, but the company who asked me to do this research, they did many subcultures. They still got very nice results. The customer was satisfied. It was the English customer. English, they like chimera petunias, was it? So he could produce 10,000 of it, and all sold a nice price. Also in the field, they were still doing very well, he told me. So it was very stabilized meristem. And we can use it for other chimeras too. If you want to maintain the chimera structure, mostly it's in the flower, 2 colors in the flower, or leaves with white margin or white yellow heart. If you have problems to reproduce in vitro because we get mixed somacronal variation, let's say, then it's better to use this topolin, especially the MEMTR. MEMTR is very anti-senescence and also keeping the meristem in a good normal division pattern.
Okay, very interesting. Yeah, Miss Bu Dwi Kusuma and Bu Nurmalasari, if you have follow-up questions, feel free to open your microphone and ask it directly to Prof. Stefaan. Okay. Otherwise, we have one more questions. Maybe one last questions because it's 5 p.m. already. Yeah. Professor, thank you for sharing your knowledge. I have questions based on your research. Which part of the leaf is the best to take for tissue culture? Is it the young one? And due to indigenous hormone inside, when is the best time to pick up the leaf from the plant? And one more question: what is the most cytokinin that you use in leaf culture? Like the most frequent, I guess.
In leaf culture?
Yeah, like
so the first is many questions, of course. So best is the young leaf, of course. It's still full of cytokinins. Yeah, the old cultures, they don't react as good. Sometimes we are a bit lazy. We take a culture from at the end of the lifespan, 6, 7. We have to avoid this. Young, good growing cultures, and you take the nice, good growing leaves is always better. I think because indeed about endogenous cytokinins available there, which can also help. So I never take leaves from the greenhouse to avoid the contamination. Yeah, but always from vitro plant. And the cytokinin I use now mostly in the vitro for propagation is the MTR, Metatopolin riboside. Very nice result with many plants. So the riboside also it helps to take up the molecule easier, the riboside. It's entering better and also transported better. But for the leaf culture, yes, nowadays we use this that combination of this inhibitors with the cytokinin to have a nice results. It's really this is the way to go, I think. Yeah.
Okay, thank you. And maybe one last questions because it's there. It's from Prof. Edi from Universitas Erlangga. To induce embryogenic callus and somatic embryo from coffee leaf, what is the best growth regulator and protocol that you will suggest?
Well, we already published this article. Maybe I can send you the link. It's all detail out there. Is, like I said, is the combination of the 2IP with the GN, that we call GNs, but it's the inhibitors of the cytokine oxidation. I will briefly look up this article and send you in the chat the link. Yeah. Yes, I have it. Just this link, you can find it. Yeah.
Yeah, we see the link.
Everything is there, and with nice pictures and concentrations. Yeah.
Okay. Wonderful. Right. Yeah, I think that's the last questions for our seminar today.
Okay.
Thank you very much, Professor Stefaan, for the wonderful talk and for your time to sharing your works, research work, and your knowledge with us today. I would like to invite all of the participants to give our huge appreciation to Professor Stefaan. Yeah.
Thank you. Of course, I would like to come to Indonesia too, of course, but okay, this may be a good way to keep it a bit feasible. Yeah.
Yes, hopefully the next seminar you will
and to meet you in person.
Yeah.
Yeah.
Yes. Yeah.
Yeah.
We hope so. Yeah. Well, hopefully next year we can invite you to give to stay here for 1 month or few weeks and to giving several lecture here.
Yeah, maybe 1 month is long, too long. Yeah. Okay. Yeah, okay, thanks a lot, and I hope you enjoy it. And thank you for inviting me. Yeah.
Thank you. I will give the session back to Miss Wirda. Thank you.
Yep.
Okay, thank you, Dr. Anjar, for guiding the guest lecture. Thank you for Professor Steven Werberg and our moderator today, Dr. Anjar, for the amazing session. And the next agenda is photo session. Please give the best smile to take picture together. And please, Ms. Farah, please help me to take picture. Okay. Ms. Farah, are you ready? Okay. Okay. I would like to remind all participants, please don't forget to fill in the attendance sheet. Ms. Farah?
Or I can take our picture if you like.
Don't worry.
If you can remove, if you can unpin, so we can have.
Yes.
If you can unpin the camera.
Unpin the camera. Okay, thank you.
Yeah. Gallery. Yeah. Okay. Wow. Okay, I will take the first slide. Sorry. Let me repeat that again. Let me take the first page. 1, 2, and 3. Smile.
Smile.
Okay, a moment. And then I move to the second page. Nobody and no camera is on, but it's okay. Just go straight. The second page. Third page. And then the last page. Okay, I'm done with the picture.
Okay, thank you, Dr. Anjar. And the last agenda is presentation of certificates to our speaker and moderator symbolically. Please, the committee help me to present the certificate. Okay, the first certificate is for certificate for Professor Dr. Stefaan Werburg. Thank you, Professor, for your amazing lecture. And the second, the next, the next, yeah. We have a problem. Okay.
It's okay.
It's okay. Okay. The next one is for a certificate for Dr. Anjar Triwibowo. Thank you, Dr. Anjar, for guiding this this lecture, the amazing session. Thank you. Okay, ladies and gentlemen, we are in the end of the guest lecture. I am SMC. I apologize for the inconvenience during the guest lecture, and thank you for your appreciation, participation, and attention, and see you in the next webinar. Thank you.
See you. Thank you, ladies and gentlemen. Thank you very much. Thank you, Prof. Stefaan. See you in the next.
It was nice. Thank you very much.
Thank you, everyone.
Thank you, Professor.
Terima kasih Bapak Ibu sudah join. Mbak Farah bisa